DNA damage and repair. In this chapter, we will explore the various types of DNA damage that can occur and how these damages alter the DNA sequence or structure. We'll then examine the cellular mechanisms that detect and repair such damage. Specifically, we will review direct reversal mechanisms, base excision repair, nucleotide excision repair, mismatch repair, and the SOS response. By the end of this lecture, you should be able to describe the causes and consequences of different DNA lesions and explain the steps of each major DNA repair pathway. A mutation is defined as any change in the DNA sequence. Mutations can range from small-scale changes, such as point mutations involving a single base pair, to larger alterations like frameshift mutations, which occur when one or two bases are inserted or deleted. These changes can disrupt gene function and lead to significant consequences at the cellular or organismal level. DNA damage occurs frequently and can result from internal processes or external agents. Endogenous damage is caused by metabolic activities within the cell, such as reactive oxygen species generated during respiration. Exogenous damage comes from environmental sources like ultraviolet light, gamma rays, and X-rays. Because DNA integrity is critical, cells have evolved robust repair mechanisms to restore damaged DNA and preserve genomic stability. Ultraviolet light is divided into three bands, A, B, and C, with UVC being the most energetic and damaging to DNA. Exposure to UV light can result in the formation of cyclobutane pyrimidine dimers, primarily between adjacent thymine bases, as well as 6-4 photoproducts. Both lesions distort the helical structure of DNA and block normal replication and transcription if not repaired. X-rays and gamma rays are forms of ionizing radiation that can damage DNA both directly and indirectly. Indirect damage: radiation is absorbed by DNA or water molecules tightly bound to DNA. In the more common indirect damage, radiation ionizes surrounding water molecules, producing reactive oxygen species such as hydroxyl radicals. These radicals can cause single-strand breaks, base modifications, and even double-strand breaks. Clusters of such lesions can lead to chromosomal rearrangements like translocations and inversions. Water itself can damage DNA through hydrolytic cleavage reactions. There are three primary types of bonds in DNA that are susceptible. The phosphodiester bond can be cleaved, though this is rare. More commonly, N-glycosyl bonds linking bases to sugars can break, resulting in abasic sites. Additionally, deamination reactions can remove amine groups from cytosine, adenine, or guanine, leading to base transitions or transversions that alter base pairing properties. When an N-glycosyl bond is hydrolytically cleaved, the base is lost, creating an abasic site. These sites lack coding information and present a significant problem for DNA polymerases, which may stall or incorporate incorrect bases during replication. Cells must recognize and repair such damage promptly to avoid mutations. Mutations resulting from base substitutions are classified as transitions or transversions. A transition occurs when a purine replaces another purine, or a pyrimidine replaces another pyrimidine. A transversion, on the other hand, is when a purine is substituted for a pyrimidine or vice versa. These changes can have varying effects on protein function depending on the position within a gene and the codon affected. Reactive oxygen species, such as hydroxyl radicals generated during cellular metabolism or by ionizing radiation, can cause oxidative damage to DNA. A well-known product is adeoxyguanine, which mispairs with adenine and can result in G to T transversions. Another product, thymine glycol, can block replication and transcription, acting as a cytotoxic lesion. These oxidative products threaten both genetic integrity and cell viability. Alkylating agents are chemicals that transfer alkyl groups, such as methyl or ethyl groups, to DNA. These alkyl groups attach to DNA bases and form adducts that distort the helix and interfere with base pairing. Many such agents are environmental toxins that require metabolic activation, often by cytochrome P450 enzymes. Some can form interstrand cross-links, which are particularly lethal because they prevent strand separation during replication. Examples include mustard gas, cisplatin, and aflatoxins. Aflatoxin B1, produced by certain fungi, is a potent carcinogen that forms DNA adducts after metabolic activation. Once activated, aflatoxin binds covalently to DNA, particularly at guanine residues. This can lead to mutations during replication and is strongly associated with liver cancer in humans. The structural distortion it causes is visible under imaging and used as a model for studying carcinogen DNA interactions. The Ames Test is a classic assay used to detect mutagens and potential carcinogens. It uses a strain of Salmonella typhimurium that carries a mutation, making it unable to synthesize histidine. When this strain is exposed to a mutagenic compound, reversion mutations can restore histidine synthesis. A high number of revertant colonies on a histidine-deficient medium suggests that the chemical is a mutagen and possibly a carcinogen. Once damage occurs, cells employ several repair strategies. Direct reversal pathways can remove some lesions without replacing the nucleotide. Base excision repair is used for small, non-distorting lesions, while nucleotide excision repair handles bulkier distortions. Mismatch repair fixes replication errors that escape proofreading. When damage overwhelms the repair capacity, cells may initiate the SOS response, enabling survival at the cost of increased mutagenesis. In many bacteria, UV-induced cyclobutane dimers can be repaired by a light-driven process called photoreactivation. This repair relies on the enzyme photolysis, which binds to the dimer and uses energy from visible blue light to cleave the cyclobutane ring, restoring the original bases. This process is specific and efficient, but is not found in humans. Another direct reversal mechanism involves the removal of alkyl groups from modified bases. The enzyme O6-alkylguanine DNA alkyl transferase specifically removes methyl groups from the O6 position of guanine. Interestingly, this enzyme can only act once. It transfers the methyl group to itself and is then degraded, which is why it's referred to as a suicide enzyme. This repair pathway is crucial in preventing mutagenesis from alkylating agents and has important implications in cancer biology and chemotherapy resistance. Base excision repair is a multi-step pathway that removes and replaces individual damaged bases, typically those resulting from oxidation, deamination, or alkylation. When a base is damaged, a DNA glycosylase recognizes and removes it, leaving behind an A basic site. The DNA backbone is then cleaved by an AP endonuclease. The resulting gap is filled by a DNA polymerase and sealed by DNA ligase. This process is conserved in both prokaryotic and eukaryotic organisms and is essential for maintaining genome integrity on a daily basis. The base excision repair pathway begins with the recognition and removal of a damaged base by a specialized enzyme called DNA glycosylase. These glycosylases come in different types depending on the lesion they target. Some are monofunctional and only cleave the bond between the base and the sugar, while others are bifunctional and also possess AP lyase activity, allowing them to cut the sugar phosphate backbone at the 3 end of the lesion. The variety of glycosylases allows the cell to respond to a wide range of chemically altered bases. Base excision repair unfolds in two major stages. The first is base excision and chain cleavage, where DNA glycosylase removes the damaged base, creating an abasic site. Then, an AP endonuclease cleaves the DNA backbone at this site. In the second stage, the repair machinery replaces the missing nucleotide and reseals the DNA. Depending on the extent of repair, the cell may use a short patch or a long patch subpathway. Both pathways ensure accurate restoration of the DNA template. The short patch subpathway replaces only a single nucleotide. After the abasic site is cut, DNA polymerase beta inserts the correct base, and DNA ligase seals the nick. In contrast, long patch repair replaces two or more nucleotides. This process uses DNA polymerase delta or epsilon to extend the new strand. As the polymerase displaces the strand ahead, a flap is formed and removed by flap endonuclease. The final nick is then sealed by DNA ligase. These two subpathways allow flexibility based on the type and extent of damage. Nucleotide excision repair targets bulky DNA lesions that distort the helical structure, such as thymine dimers and large chemical adducts. This pathway operates by removing an entire oligonucleotide segment that includes the damage, typically 24 to 32 nucleotides in length. The general steps are conserved in both prokaryotes and eukaryotes. The lesion is first recognized. Then endonuclease cut the DNA on both sides of the damage. The damaged fragment is removed, and the resulting gap is filled in by DNA polymerase using the undamaged strand as a template. Finally, DNA ligase seals the strand to complete the repair. Eukaryotic cells employ a nucleotide excision repair system that closely resembles the bacterial version, but involves a larger and more complex set of proteins. In humans, defects in this pathway are associated with the genetic disorder xeroderma pigmentosa. Patients with this condition are extremely sensitive to UV light because their cells cannot repair thymine dimers, leading to high rates of skin cancer. This clinical correlation underscores the importance of proper nucleotide excision repair in maintaining genome stability and preventing cancer. Mismatch repair corrects errors that escape proofreading during DNA replication. These include base mismatches and small insertions or deletions, often resulting from polymerase slippage. While DNA replication is highly accurate, mistakes still occur at a low frequency, and mismatch repair provides a secondary safeguard. Without it, replication errors would accumulate and significantly increase mutation rates, leading to genomic instability and potentially cancer. In E. coli, mismatch repair is distinct from that in gram-positive bacteria and eukaryotes because it uses methylation to identify the newly synthesized strand. The parental DNA is methylated at specific GATC sites, whereas the daughter strand is initially unmethylated. The repair machinery can thus recognize which strand contains the error. This methylation-guided discrimination is critical to ensuring that the correct base is retained during repair. The repair process begins when a mismatch is recognized by the MUTS-muddle complex, which activates the MUF endonuclease. MUF cleaves the unmethylated daughter strand near the GATC site. Exonuclease then degrade the segment containing the error, creating a gap. DNA polymerase III holoenzyme fills in the gap using the parental strand as a template, and DNA ligase completes the repair. Eukaryotic cells use a similar overall mechanism, though they rely on different strand discrimination signals since they lack methylation marks. Mismatch repair not only fixes replication errors, but also helps preserve microsatellite stability. These short, Repetitive sequences are prone to slippage during DNA synthesis. A defective mismatch repair system leads to microsatellite instability, a hallmark of certain cancers such as hereditary non-polyposis colorectal cancer. By removing mismatches and correcting small loops, this repair pathway plays a central role in safeguarding the genome against replication-induced mutations.
Although rare, incorporation of base pair mismatches during DNA replication does occur. There are DNA repair mechanisms that can fix such mistakes. One such mechanism is the methyl-directed mismatch repair system. In Escherichia coli, this repair system can detect a mismatch shortly after DNA replication and repair the newly synthesized strand, thus preserving the base pairing that was present in the original DNA molecule. The system relies on the fact that methylation of a newly synthesized DNA strand does not occur until several minutes after the strand is made. This provides a window of time during which the newly synthesized daughter strand in a DNA double helix can be distinguished from the parental strand, which is already methylated. A sequence of DNA with only one methylated strand is said to be hemimethylated. Actual recognition of a base pair mismatch is the role of the mut-S protein. Mut-S binds to a mismatch site and forms a complex with another protein, mut-L. The formation of this complex recruits mut-H to the region. Mut-H binds to a hemimethylated site. The mut-S, mut-L complex interacts with mut-H by a DNA looping mechanism. Mut-H then makes a cut specifically in the non-methylated strand. An exonuclease digests the non-methylated strand from the cleavage site to just beyond the mismatch site. This leaves a gap in the daughter strand which is filled by DNA polymerase III and then sealed by DNA ligase. The end result is that the mismatch has been replaced with the correct base pair as indicated by the original DNA strand.
When comparing base excision, nucleotide excision, and mismatch repair, it's important to note their specific roles. Base excision repair is suited for small, non-distorting lesions like oxidative damage or deamination. Nucleotide excision repair handles bulky, helix-distorting lesions such as thymine dimers and chemical adducts. Mismatch repair fixes replication errors that escape proofreading. While each pathway has its own mechanism and specialized proteins, all three are essential for maintaining genetic fidelity and preventing disease. When DNA damage is too extensive to be repaired quickly, such as from abasic sites or thymine dimers, the replication machinery can stall. This stalling can be lethal unless the cell activates a tolerance mechanism. In E. coli, the SOS response enables the cell to bypass damage by expressing a set of specialized, error-prone DNA polymerases. These enzymes can synthesize DNA directly across damaged regions, allowing replication to continue, but at the cost of increased mutation. This trade-off ensures cell survival under genotoxic stress. The SOS response is repressed under normal conditions by LexA, a protein that binds to the regulatory regions of SOS genes. When DNA damage occurs, single-stranded DNA accumulates and activates RecA. RecA forms a nucleofilament and acts as a coprotease, promoting the self-cleavage of LexA. This de-represses the SOS genes, allowing expression of error-prone polymerases and other DNA repair proteins. The coordination of LexA repression and RecA activation is central to initiating the SOS response. During the SOS response, E. coli induces several DNA polymerases. DNA polymerase II is a high-fidelity enzyme that helps restart stalled replication forks. Polymerase IV binds to the sliding clamp and takes over synthesis when DNA polymerase III is blocked. Polymerase V, activated by RecA, is the main translesion polymerase. It is capable of synthesizing DNA across a wide variety of lesions, including thymine dimers and abasic sites. While these enzymes are more error-prone, they provide a temporary solution that allows the cell to complete replication and divide. Eukaryotic cells also use translesion synthesis to tolerate DNA damage. Humans possess at least 14 specialized DNA polymerases, some of which participate in this process. Each of these polymerases has a unique substrate preference and can bypass specific types of DNA lesions. For example, polymerase eta is particularly effective at bypassing UV-induced thymine dimers and is deficient in individuals with xeroderma pigmentosum variant. The redundancy and specialization of these enzymes allow eukaryotic cells to survive DNA damage with flexibility, albeit with an increased risk of mutations. Timeline to show the selected key milestones in DDR mechanism identification, DDR correlation with carcinogenesis and drug discovery. As early as 1775, the linkage between cancer predisposition and environmental insult was observed. However, until 1940s to 1960s, the correlations of carcinogenesis and DNA damage induced by chemicals or radiation became clear with the emergence of molecular biology. Since 1970s, DDR pathways have been depicted as a spectrum of catalytic processes, protein-protein interactions, and protein-DNA interactions. Deficiencies in DDR pathways could facilitate carcinogenesis and can be targeted by small molecule drugs, such as PARP inhibitors approval for the treatment of BRCA mutant ovarian cancer. All these efforts eventually led to the grant of Nobel Prize in Chemistry in DDR area in 2015. Now, a great deal of interest has been evoked for the study of DDR mechanisms as well as anti-tumor drug discovery.